Hello, everyone. My name is Ivan. I'm a final year biochemistry PhD student in Andy Fire's group here at Stanford, and I'd like to give you an introduction to Obelisk RNAs, which are this group of weird subviral RNAs that seem to show up in microbiomes all over the place. And this work was made possible by a collaboration with Robert and Maria and Marcos and Artem and Ami Bart and her group and a bunch of other collaborators. So, kind of to set the stage, I'm personally very interested in RNA viruses because they're very simple genetic systems, yet they're so diverse, and they're so diverse in their diversity. But one thing that unifies RNA viruses almost by definition is that no matter what you are, whether you're the very simple, very small MS2 phage of E. coli or the devastating influenza A virus, you have to encode these enzymes for the replication of your genome, these so-called replicative polymerases. So there actually exist entities that are simpler than this. They almost behave like viruses, but they don't have this requirement. And to me, that's fascinating. And most exemplary of those elements are so-called viroids. So viroids are these tiny infectious RNAs that have these highly structured genomes depicted here in these skeleton diagrams. And they're these small circular RNAs that don't code for any proteins, and yet they cause economically devastating diseases in crop plants around the world. On the human side, there's hepatitis delta virus, which is a hepatitis-causing virus that can co-infect or super-infect a person who's already suffering from a hepatitis B virus infection. And this can cause much worse disease and actually can lead to liver cancer. And so this is also a small circular RNA. It's about 1,500 nucleotides in length. And here's a depiction of the genome as a skeleton diagram, but also throughout this talk, I'll be using these so-called Jupyter plots, which you can read like a plasmid map where chords represent predicted base pairs and the circumference is the genome coordinate. So hepatitis delta virus does code for a protein, but it's not replicative machinery. And so these elements really fascinate me. But when I kind of started on this project, 2020, 2021, roughly speaking, these three types of elements were kind of it for subviral circular RNAs. And that was really striking because back then, today, and it seems to be continually growing, our knowledge of the diversity of RNA viruses just keeps growing. Like last year, the number of RNA viruses literally grew by 1 million distinct sequences. And yet this number of subviral RNAs didn't really change. So I wasn't the only person who was interested by this. There's been lots of really interesting recent work on this, and I particularly point you towards this one from the Weinberg group, and also this one from Eugene Koonin by Ben Lee, trying to address this discrepancy of like where are these subviral RNAs? We're seeing so many viruses, but not as many as we'd expect for subviral RNAs. And so the way I approach this is I created a bioinformatic tool called Viroid Nominator, or Venom. And what it does is it takes RNA-seq data and it gives you contigs that are consistent with being viroids. They're not guaranteed to be viroids, but it's just a crude filtering tool. And slightly different to how the other papers did their searches, but I still highly encourage you to check out Ben Lee's work from last year. And if you're interested in looking for viroids in your own stranded RNA-seq data, then please check it out on GitHub. Suffice it to say that Venom works fine, good enough for me. And so I decided to look for viroids or viroid-like agents in microbially enriched data. And probably not a surprise to the audience here, certainly not Dr. Kate Bowie, who just mentioned him, but Curtis Huttenhauer's group has generated a lot of really useful resources, and this is the resource I started with by Jason Lloyd Price. It's really careful RNA-seq of human stool samples gathered over a year in some cases, all gathered around in 2013. And very quickly, when I started analyzing this data, this thing popped out. So this really struck us because of its almost perfect rod-like predicted secondary structure. It reminded me of Cleopatra's Needle in London, which is where I went to school. And so ultimately, we ended up calling them obelisks, and this is the first obelisk, so it's obelisk alpha. And beyond this like crazy rod-like secondary structure, like if you take a random mRNA and predict its secondary structure, it doesn't look like this. So beyond this crazy secondary structure, what was also striking is that this reasonably short RNA had no sequence similarity to any database that we could think to look in. So it seemed to be like this completely unannotated RNA. And it wasn't just a one-off. It actually showed up in multiple donors in this Hutton-Howard data set. And because it's longitudinal data, we could actually see that these obelisk alphas were actually persisting within specific donors. And what's more, each donor seemed to be accumulating donor-specific SNPs. So my obelisks would be different from someone else's obelisks, despite both being the alpha obelisk. So really evidencing potentially colonization of the human gut microbiome. So to cut a really long story short, they also, obelisks, show up in pure bacterial cultures. And so my advisor, Andy, found this. The oral microbe, which is really interesting to hear Laura Waver talk about this, Streptococcus sanguinis, harbors an obelisk. And fortunately, Cleopatra had 2 needles. There's one in New York, and it also has these similar features: very rod-like predicted secondary structure, about 1,000 nucleotides in length, and again, the sequence doesn't resemble anything we've seen before. So, as we started to accumulate examples of obelisks, we were able to then do homology or sequence similarity-based searches across lots of RNA-seq data sets. And in the SRA, where we did most of our searches, the data are overwhelmingly biased towards Western microbiomes. So that's what we ended up sampling. But these obelisks seem to show up in Western microbiomes. So looking over about 440 different donors' samples, about 7% of people harbor at least one obelisk. And even more strikingly, about half of the mouth samples that we were able to analyze harbor an obelisk, though there's very few mouth metatranscriptomes publicly available in the SRA, so that number probably will change. So now we have like this large collection of different human-associated or microbiome-associated obelisks. We can actually start sketching a kind of genome plan. And so all of these elements are about 1,000 nucleotides long, apparently circular, and highly base paired, making predicted secondary structures that look like rods. And they also seem to segregate by anatomic sites. So the obelisks you find in the mouth are pretty distinct from the obelisks you find in the gut, despite those two areas of the body being connected. And just to really reiterate the weirdness of the secondary structure, hepatitis delta virus folds in half, but its open reading frame, the hepatitis delta antigen, is on one side of the hairpin. Whereas in obelisks, the largest open reading frame, which we call Oblin 1, is actually self-complementary. So this is a protein sequence that has to code both for a protein sequence and a self-complementary genome. So that the constraints on sequence space must be crazy and super interesting to study. So this Oblin 1, we have no idea what it does. AlphaFold gives a pretty confident prediction of an N-terminal globule, but actually it's not very well conserved across the sequences that we've seen. The sequence diversity in Oblin 1 is really great. And actually the most conserved part of the protein, AlphaFold doesn't predict to fold in a tertiary structure. So it makes it very difficult for us to guess what this might be doing. But given that this conserved, quote, domain A is enriched in positive residues, we think it might be interacting with RNA or other proteins. And these are things that we're following up on. And so kind of to summarize, obelisks are, we ended up finding about 30,000 obelisks showing up in 200,000 different data sets. They seem to be a phylogenetically distinct group that are related to one another. So you can attempt to estimate a phylogeny, but because of the crazy sequence constraints on the protein, I reckon this tree will change a lot over time. And they show up on every continent. And obviously, where they show up is very biased by what continents are able to contribute large amounts to the SRA. But they do show up on every continent, including Antarctica. So, yeah, 1,000 nucleotide RNAs that we previously hadn't cataloged, they seem to be kind of in every microbiome, basically, that you look in. They have this crazy unknown protein, which we call Oblin 1, that has like this conserved sequence, but we don't know what it does. And obelisks appear to persist and maybe even speciate in people. And we see that there might be some kind of mammalian bias, but that also could be due to the nature of how the searches were done. And there might be a bias towards the mouth as well. But again, that could be an alias due to the data that we have available. And so with that, I'd like to thank everyone who made this possible, as well as the funding that allowed us to do this, and the organizers for allowing me to present. So thank you.